Hello, this is Mrs. Saunders. I'm going to do the video to go over genetics notes 1 on Google Slides now. So we're going to start out here. That's a great picture of DNA. We are not going to jump right into DNA, actually. We're going to start with Gregor Mendel. He's the founding father of genetics. He was an Austrian monk. Austrian monk who studied pea plants in the 19th century. It was his job to tend to the gardens at the monastery. He was a scientist. He took tons of data. He focused a lot of his work on pea plants, looking at the different colors, the pea pod shape, size, all sorts of stuff about the pea pods. He ended up developing these 3 laws. The law of dominance. A dominant allele masks the effects of a recessive allele. We will talk more about that on the next couple of slides, along with the law of segregation. Chromosome pairs separate individually into gametes, into the sperm and egg cells, to transmit genetic information to offspring, the law of segregation. You learned about this last unit in meiosis, and then we went on to it a little bit in reproduction. And the last law is the law of independent assortment. Alleles for genes assort independently of each other. Here's a little picture of some of Mendel's pea plant crosses that he worked on. He looked at the different, whether it was a round shape, had a wrinkle to it, different colors, the pod coat, whether it was smooth or constricted, if it was tall, if it was short. He literally took what you learned last unit with reproduction, took the stamen, the pollen, and he would self-pollinate the flowers on the pea pods. So, he would cross-pollinate like you learned about right on top of the pistil and look at his offspring. There are quite a bit of genetic definitions that we got to start with to give you a good foundation. A gene is hereditary material found in the chromosome. That determines the trait. The allele is the different forms of the genes. The dominant is the allele that shows up. We use uppercase letters to represent dominant. For recessive, we use lowercase letters. The allele that is present but not expressed, the recessive. Genotype. This is the genetic makeup of an individual that's represented by a letter, one from each parent. So you get 50% from mom and 50% from dad. You learned about that with meiosis. 23 chromosomes from mom and 23 from dad. When you go to write a genotype, it does not matter which one you write first. Okay. So in this example, if mom or dad, it doesn't matter who gave you what. In this case, they're both dominant. So you write 2 uppercase letters. In this case, mom and dad both gave you the recessive. So you write 2 lowercase. But in this case, one of your parents gave you the dominant and one the recessive. Technically, if you wrote the little a first and then the big A, we wouldn't mark it wrong. But when it's the mix, it's preferred that you put the uppercase letter first. Phenotype. See the word P, the 2 letters PH? Phenotype is your physical characteristic. So it's genetic traits that are expressed as the physical appearance. So we want to see words when we ask you for a phenotype: green, yellow, tall, short. When we ask you for a genotype, we want to see the letters. The geno, see the word gene, the combination. 2 uppercase, 2 lower, or the mix. Now, homozygous and pure mean the same thing. Both alleles for a trait are the same. So it's either going to be 2 uppercase or 2 lowercase. If you see the word heterozygous or hybrid, that means the same thing, a mix. Alleles for a trait are different. So you see the uppercase and the lowercase. The P generation stands for parents. The F1 generation are the offsprings. It's Latin for philo. That's the kids, the offspring. Punnett squares are those boxes. You did do them in 7th grade. Some kids remember them and it comes very easy. Other kids have a harder time with this. You will see I attached a quick little 3-minute video to go over it. But Punnett squares are diagrams that show the result of a cross between individuals. We'll look at some Punnett squares on the next couple slides. A test cross, that's used to find out if an individual showing the dominant trait is homozygous or heterozygous. So someone looks dominant. Why would we do the test cross? To see what their genotype is. We always cross somebody that looks dominant with a pure recessive. If they end up having an offspring with the recessive, then we know they were the hybrid. Recombination, the appearance of genetic types in the second generation that were not seen in the first. So if both parents were the hybrid, let's say tall is dominant to being short, so big T is dominant to little t, both parents are hybrid, and the offspring is short to little t's, that would be recombination. Gene linkage. This is when traits located are on the same chromosome tend to be inherited together. Red hair and freckles are an example of this. They are right next to each other, so we say gene linkage. Now you did learn though, gene linkage, genes can be separated by crossing over during meiosis. When you saw those two little chromatids talk to each other and exchange DNA segments. This is how to use a Punnett square. You would put one parent on the top and one on the side, and it doesn't matter which side you pick. Then you take the gene and you carry it across, just like the arrows show, all the way across and both sides. There's an example of one. Now, you also have to calculate the genotype and phenotype of Punnett squares. This is an excellent video. Just you go ahead and you click on it. It's 3 minutes. Go ahead and watch it. We will not do this right this second, but this is what a problem will look like. In roses, having thorns is dominant to not having thorns. Use the Punnett square to predict the probability that the offspring produced between two heterozygous roses will have thorns. So you go ahead and you put one parent on one side. Okay, the hybrid, two heterozygous roses, big T, little t, big T, little t. You bring that big T down, the little t down, the big T goes across, the little t goes across. Again, if the little t is in front of the big T, it will not be marked wrong. It's just proper to put the big T first. So genotype, you want to list all your possible genotypes. So I've got big T, big T, big T, little t, little t, little t. This is how you say it in words. You don't have to have that there, but you have 25%, 50%, 25. So 25, these two make up the 50, and there's the 25. You can also do it in a ratio, 1 to 2 to 1. You always want to make sure you answer in the way the question asks you to. Phenotype, PH, think pheno, what it physically is. This is words. So when it's the hybrid, when it's simple dominance type of inheritance, which is the only type I've gone over so far, when it's the hybrid, it looks like the dominant. So these 3 boxes are all dominant. That's how I got the 75% thorns. There are no alleles that you have to have memorized. They have to give you the given up here in the problem. So in roses, having thorns is dominant. They have to give you that. That's how I know the capital T is having thorns. So these 3 boxes are dominant. 25% no thorns. That would be the pure recessive or the homozygous recessive. Simple dominance is the type I've gone over so far under the different types of inheritance. One dominant allele is expressed over a recessive. Now, this is something different we haven't talked about yet. Incomplete dominance. In some cases, the 4 o'clock flower, red flowers and white flowers actually make pink. And here's a nice picture of them. This is similar but different. In this type of inheritance, it's called codominance. Neither allele is dominant over another. So both red and white hair appear. So if you look closely, there's some white and there's some red. A great example of this is roan cattle coat. You can see both red and white on the cattle. Sickle cell anemia is also like this. The last type of inheritance on this page are lethal genes. Some combinations of alleles cause the embryos to die. Pure dominant white horses, pure dominant, so uppercase, WW are lethal. But think about this, there's white horses out there. So what must all those white horses be? The heterozygous. They are the hybrid, the mix, capital, lowercase. Here's a graphic of the incomplete and codominance on a Punnett square showing the red and white. In this case, you can use all capital letters. That's fine because you're getting the mix, the pink, RW. Same with codominance. We're fine if you write it like this, RW. There's your white heifer at the top with your red bull. And there's a great picture of a roan coat. Another type of inheritance are multiple alleles. Some traits have more than 2 alleles. This is important. Blood is often on the regents, so let's make sure you really understand multiple alleles. These traits are inherited by both simple and codominance. So you've already learned for blood type that there's 3 different alleles. We've already taught you this. That A has the antigen, the A marker, remember, around the cell membrane. So type A blood has that A antigen on it. Type B blood, remember, had like a B marker around it on the receptors. O had nothing. Remember O, when we would draw it, there was no receptors. That's why they're, remember, the universal donor. So both A and B are both dominant. I made that bold, over O. But neither A or B are dominant over each other. Let me repeat that. A and B are both dominant over O. So O is like a little letter to them. But neither A or B are dominant over each other. So in my way of thinking, let's look at this first. A and B are both dominant over O. So if both parents give an A, that's an easy one. That's two uppercase A's, type A. If one parent gives an A and the other parent gives an O, see how I wrote it in lowercase? Just kind of so you would get it in your head that is recessive. That would be type A because O is recessive to A. Let's say both parents give a B. So remember, they're codominant. They're both uppercase. That's type B. And then let's say one parent gives a B and an O. Remember, O is recessive. So I'm writing it lowercase to try to get you to understand it. So that would be type B blood. B is dominant to O. Now, if both parents give a little O, like a recessive, think of it like that, that's all they have. So that has to be type O. Both parents, or one parent gives an A, one parent gives a B, they're both uppercase because they're both codominant. That would be the blood type called type AB. And again, these are phenotypes. These are words, okay? Now, the way I have written it here always makes sense to my students, but that is not how they write blood genotypes. We got to go over to this third part. It has to do with Latin. They use uppercase I and superscripts. So just try to ignore the I's. Just look at the superscripts. Uppercase AA. Oh yeah, that's the one where each parent gives the AA. That would be type A. Now this parent gave an A, but see how this is a lowercase italics I? That represents the O being recessive. They do not use a superscript. So if you see an I with a superscript A and a lowercase I, that's the AO one, the hybrid for type A. This one is written like this, type B genotype when it's the pure or homozygous, 2 uppercase Bs. Now, how did we write the other one? Look up here. We use that lowercase I. So when B is the hybrid type B, you're going to do that lowercase I representing the O. So type O, the genotype is written just 2 lowercase i's. And then type B is superscript A. Oops, superscript B. I got to add that in. And the last part here is polygenic traits. Let me see if I can. Oops. Polygenic traits down here at the bottom. Traits determined from multiple pairs of genes. Color, height, eye color, fingerprint pattern, multiple pairs of genes determine traits. It's not always so simple as tall as being dominant over being short. All right, that's it for the notes for the first one. Notes, slide 1.